Hi girls, it's Mrs. Vasquez. Welcome to my recorded lesson, and here we're going to be looking at section 10.3. So if you want to have your textbook out, I would recommend maybe flipping through the pages as I'm going through the notes. We're going to be going through about 3 slides, so if you don't have your binder or your notebook with you, I would pause this video and go grab it so you can take your notes. So up until this point, we've been talking about the cell cycle. We've been talking about what happens in a cell's life from the minute that it's created through mitosis, as it goes through its own life until a time where it's ready to divide again. So if you look here, just a quick reminder, below in this picture we see, actually let me just make this a little bit longer, can I? I don't think I can, but you're not missing too much. Okay, so in this picture over here, we're looking at a very similar picture. You'll see here, if you were to think of this as maybe a pizza pie, the part of the pie that has the gray area in the perimeter, that is the main phase of a cell's life, which is interphase. And this just means the cell is not dividing. So its DNA is in the form of chromatin, it's just growing, its organelles are just doing their daily activities. And this stage, interphase, is so big that we divide it into 3 subsections. And you can think of those sections as a beginning, which is G1, a middle, which is S phase, and then the last part of interphase, which is another period of growth, which we call G2. And then, of course, when a cell completes interphase and is ready to undergo division of its own, it's going to enter the second and shortest phase of its life, which is mitosis. So up until this point, we've been talking about how it does that and what happens as it's doing that. But now let's ask the question, why does it need to do that? And how does the cell know when it's time to go from interphase to mitosis? Or when it's time for a cell to go from G1 to the S phase, or from G2 to the beginning of prophase? And that is going to be the topic of the beginning of section 10.3, which is called regulating the cell cycle. And what we see here in this picture that's different from the earlier picture we've looked at is the presence of these terms called checkpoints. So here, if you look carefully, you'll see 3 checkpoints. Over here where my mouse is the G2 checkpoint. Here we have the M checkpoint. And over here on the bottom, we have the G1 checkpoint. Over the course of a cell's cycle, it has 3 different checkpoints, and they work really similarly to maybe checkpoints we'll use in our actual lives. So I'm sure all of you guys at one point have been maybe driving with your family, and as you get off the highway on the exit ramp, you might run into a checkpoint. So sometimes police run checkpoints where they're kind of stopping each car as they go, and for a multitude of reasons. Sometimes it's to look for a particular car or person, sometimes it's just to check for seatbelts. But the purpose of these checkpoints is to stop, take a look, and then if you pass all the requirements, you have your seatbelt on, you're not the car in question, you get the green light and you can go. Or if you don't pass all the requirements, then you'll be held up and you'll be stopped. And the checkpoints in cells work the same way, and they're regulated by a type of protein called cyclins. So cyclins are a group of proteins. If you think back, so a quick March review, if you think back to proteins, remember proteins are a type of organic molecule like carbohydrates and lipids. Some proteins, remember, speed up chemical reactions. Those would be called enzymes. Cyclins, however, are proteins that live in the cell, and what they do is they control these checkpoints. So they're the ones deciding whether the cell can pass the checkpoint or whether they are not ready to pass the checkpoint. So they're involved with regulating the cell cycle. So they'll tell the cell a couple of things, and it really depends on which cell checkpoint you're at what specific time. So for example, we say over here, they tell the cells when it's time to divide. Knowing when it's time to divide, that's going to happen in the G2 phase. So what's going to happen is if a cell's in G2, that means it went through G1, it grew, it duplicated its DNA in the S phase. And then in G2, what those cyclins are going to do, they're going to stop, they're going to take a look and see, okay, was the DNA replicated properly? Are there any mistakes? Is the cell growing? Are the organelles doing their job? Is everything running smoothly? And if the answer is yes, then they'll give the go-ahead. They'll turn the green light on and they'll tell the cell, okay, now it's time you can enter mitosis. Same thing with the G1. Did the cell grow properly? Can it get ready to do mitosis and start duplicating its DNA? The M checkpoint, however, that's the only checkpoint that's in mitosis, and its job really is just to make sure mitosis is going well. If, for example, prophase, metaphase, anaphase has been working properly and the chromosomes have been evenly split between the two cells, then the cell will pass that M checkpoint and it will be allowed to finish and complete mitosis. Okay, so this is a really important part of cell life, knowing when they're allowed to pass through different stages so they can progress through their life cycle. The next part of this section, though, is talking about a special small group of cells that for some reason don't have the ability to listen to these checkpoints. These are bad cells. These are not good cells. You don't want cells that don't listen, but unfortunately there are a group of cells that we've found over the years have an inability to listen to cyclins, and they don't follow checkpoints. So for example, if a cell was not supposed to pass a checkpoint because it didn't meet the requirements, these types of cells would go ahead anyway. And so these cells, which you've all have heard before, are cancer cells. So that's the definition of a cancer cell. Unfortunately, you probably know someone who has had cancer or might have cancer, so we're aware that there's many different types of cancer. But the one thing they all have in common is that one of the reasons that makes them so dangerous is that they have the inability to listen to regulation. So they don't follow directions from the cyclins, they don't follow directions from checkpoints, and so we say that cancer cells have uncontrolled cell growth. And cancer usually begins with the production of a tumor. So we probably all have heard of the word tumor before. All a tumor is a mass of cells. It's a group of cells that don't stop dividing, and so they produce this mass. And tumors actually come in two different forms. And you've probably heard of these types of tumors before. Out of the two, there's one that's considered a little bit better to have than the other. It's the one that's non-cancerous, and it's the one that's a little bit more easily treatable. Usually it's fairly easy to remove them, whether through surgery or through procedures. And those are called benign tumors. Like, for example, if anyone here has a wart or maybe has had a wart removed before, warts are known as benign tumors. So again, not really dangerous. They're easily taken off. Some people don't even remove their warts because they grow, so they don't follow regulation and they overgrow. But at some point, benign tumors stop after a while and they don't spread, which is a good thing. However, there are a group of tumors that unfortunately are much more dangerous, and these are the tumors that are cancerous. So we refer to them as malignant tumors. So for anyone who's interested in root words like I am, mal in malignant, that means bad. So malignant tumors are the worst type of tumors. These are the ones that are cancerous, and what makes them so dangerous is that not only do they over-duplicate and they keep dividing, but they have the ability to spread to other parts of the body. So these cells, these tumors, they grow so much that sometimes part of it breaks off unintentionally, and maybe the blood system picks up that tumor, transports it to a different part of the body, they'll land somewhere and they'll keep growing. And so if you've ever heard of someone maybe talking about cancer, you might have heard them refer to the cancer maybe spreading or not spreading. People who are diagnosed with cancer usually hope that they've caught the cancer early enough, because the earlier you detect your cancer, if you're diagnosed with one, the less of a chance that it has spread already, which means it's easier to treat. So we definitely don't want cancer spreading, but if it does spread, there's a term for that, and it's called metastasis. So let's see. Now I realize you might not see the bottom. Hold on one second. Let's see if I can move it a little bit lower. Okay, that's better. Okay, so the spread of cancer cells beyond their original site is called metastasis. So if someone said the cancer has metastasized, that usually means the cancer has moved beyond the original tumor, the original site, and has spread to a different part of the body. And again, this is not a good thing because it usually just means it's harder to treat because you have to look at multiple areas of the body at once as opposed to just treating one smaller original site. So metastasis is spreading, but the next thing we'll be talking about is causes of cancer. So one thing that all cancers have the same is their inability to follow cell regulation, cyclins and checkpoints, and their cause, even though the causes might be slightly different, what they all have in common is that it has something usually to do with a defect in the gene. So that means a defect or damage in the DNA of the chromosomes. And usually it's a gene that's responsible for regulating cell growth and division. So if there's a defect in it, that usually means there's a problem with cell growth and division, leading to it becoming a cancer cell. So what are some causes of these genetic defects? Well, I'm sure we've all heard that smoking is really bad. Well, it's bad for a number of reasons, but one of the big reasons why smoking is bad is because it's been very highly linked to higher rates of lung cancers and some other cancers, but mostly lung cancer. So between the chemicals that are in, the nicotine that's in the cigarettes, something about those chemicals has increased defects in the gene and has been correlated with higher risk of lung cancers. Then we also have radiation exposure. So radiation can include a number of things. You might remember, quick throwback to photosynthesis, remember the electromagnetic spectrum, where radiation is high energy waves. And the higher and stronger the energy, the deeper it can infiltrate your skin and go into your DNA and mutate it. So things like UV rays, that's why we protect ourselves with sunscreen, or x-rays. That's why they cover the parts of your body at the doctor that don't need to be x-rayed, and they only leave open the part that they actually want to take photographs of. And of course, if you go to get an x-ray once in a while, that's not a big deal. This is more for people who are exposed to radiation constantly. Next one is other defective genes. So what we'll find is that some genes can actually impact the effect of other genes. We're going to learn more about genes in the next chapter, because our next chapter is genetics. But a gene, just so you know, is just a part of your DNA. It's a particular segment of your strand of DNA that codes for a particular trait. For example, in order to determine what kind of eye color you have, the instructions for what eye color you have are in a particular gene. And then the gene, let's say for your blood type, the instructions for what blood type you are, that's in a different gene. So genes just code for different things inside your bodies. And the last one is something called the P53 gene. So scientists are still trying to figure out what exactly the P53 gene is, but what we know so far is the P53 gene is a gene that's found in in everyone's body, and it's a gene that for some reason, when we study the DNA of people who have cancer, for some reason this particular gene, there's always something wrong with it. It's defected in some way, whereas people who don't have cancer have a perfectly healthy P53 gene. So scientists think that something about the damage to P53 increases highly the risk for cancers and tumors. So P53 gene is just a particular gene that we think has some sort of correlation to cancer. When you have it and it's perfectly normal, you have lower risk for cancer. But when there's damage to it, we call it a tumor suppressor gene. When there's damage to it, that ability to suppress tumors isn't there, so that increases your risk for cancer. Okay guys, so that's it for today's lesson. Again, this is going to be up on the website until you have your test. So it's not going to go away. So you can come back to this and read. You can pause the video, go back to it. Make sure you have the notes of these slides taken down. I'm going to check them in class on Wednesday. And when we come back on Wednesday, you'll have a chance to ask any questions. We'll go over some of these topics, but you'll have the notes written down so we can actually move forward and finish cancer on Wednesday and then move into our last type of cell division, which is called meiosis. So meiosis is going to be very similar to mitosis, but a few big differences, key differences. Okay, so make sure the notes are written. And again, I'm sorry for the length of this video. I think I told you guys in class it was going to be much shorter. Guess I'm talking too much, so didn't mean for it to be so long. But I'll see you guys all on Wednesday. All right, bye-bye.